Hello, everyone. My name is Jared Richardson, and I am a second-year mathematics PhD student at the UT Arlington. Today, I'm excited to present some research previously completed titled Bayesian Networks with Applications for Analyzing Causal Relationships in Rice. This research was done in collaboration with researchers at the USDA ARS, Dale Bumpers National Rice Research Center, and I have very many thank yous to them. Of course, rice serves as a great model species for other grass and cereal crops, but moreover, rice is one of the most important crops in the world, feeding over half the global population. But growing it efficiently and sustainably, especially under changing climate conditions, requires understanding of how genetic markers and observable traits interact. Our goal is to build causal models that go beyond the traditional correlation analysis. And from a mathematics perspective, using statistical inference and probability, we can learn relationships between variables. We can begin with something familiar, such as a Pearson correlation coefficient. Of course, you don't need to be a mathematician or a scientist to remember what this is, but in case you forgot, this is simply a measure of how two variables linearly covary and can take on both positive and negative values. In any case, one thing's for certain, it does not tell us what variables cause the other. To explore this, we need to look at directed acyclic graphs, or what I'll be referring to as DAGs. These are graphs with specific characteristics. Three in particular are the nodes, representing variables. These things can be like traits or genetic markers. Additionally, it consists of directed edges, showing the direction of potential influence. And finally, the acyclic characteristic, meaning we cannot have circular logic or no loops are allowed within our system. We can extend this DAG into a Bayesian network, which gives us a full probabilistic model of all variables in the system. Mathematically, a Bayesian network represents a joint distribution. Each VI is a variable in the system, such as a trait or a single nucleotide polymorphism marker, and parents of VI, which refer to the nodes which point into the node VI. So the formula essentially says the probability of the whole system equals the product of the conditional probabilities of each variable given its parents. Or in simpler terms, we can break each complex multivariate system down into simpler local pieces, each representing a conditional relationship. But before we can start building these great networks, we're going to need some nice data. To do this, we're going to process noisy traits data from field experiments. This is where linear mixed modeling comes in. Y denotes the observed trait value, such as grain yield for a given rice plant. X beta being our fixed effects, things such as temperature, soil quality, or irrigation system. And finally, ZU, which represents random effects. These represent the variation between different genetic lines or environments. But why use this model? Quite simply, it helps us separate signal from noise. From this, we can compute best linear unbiased predictors, or BLUFs, which give us nice, consistent estimates of each trait, accounting for environmental effects. So essentially, you can think of it as a batting average. It tells us how well a trait performs after normalizing for external factors, and these are the values in which we will use for our Bayesian network. So once we have trait values, we can use a hill climbing algorithm to construct the network. This is an optimization process starting with an empty graph in which we iteratively add and remove edges to maximize a scoring function or likelihood function. We can apply constraints like preventing single nucleotide polymorphism markers from predicting each other. Additionally, we can introduce these priors to keep the graph biologically reasonable. For instance, we can enforce a genotype to cause a phenotype, but not the other way around. Then we estimate the parameters of the network, that is, the actual values of each conditional probability. And to do this, we utilize k-fold cross-validation. This means we divide our data into folds, train on some, test on others, and repeat. And after this is all done, we get nice graphs that look like this. This is only a small subset of our traits, as the DAX can get quite large, but this one here is using the agronomic traits from our rice population. Each node represents a trait or SNP marker based on colors. Arrows show directional influence, and then edge colors show correlation strength and population-specific variation. For instance, relationships may look different in one rice variety versus the other. But essentially, this graph allows us acts as a causal roadmap or a visual summary of how traits and genes might influence each other and be grounded in real data. Thank you for listening, and bye.